Hi, good afternoon, everybody, and welcome to Curiosity Club, the first in what we hope will be many new family afternoons of fun and discovery. We're coming to you at least in theory from the Wellcome Genome campus which is just based south of Cambridge. For those of you who maybe aren't from the area. We're a site of scientific research. We have hundreds of scientists working at the Wellcome Genome campus doing lots of really exciting research, and it's some of that research that we're here to share with you this afternoon. It's lovely to see so many of you joining. I know we have other people coming in, but we've got such an action-packed schedule that we wanted to get started without delay. Just a few very quick things from me before I hand you over to my colleagues who'll tell you much more exciting things. I have some of the basic things to help you get the most out of using this Zoom space. I'm sure many of you are familiar with Zoom by this point in 2021, but you will see us coming up on screen and hear from us. We can't see you, sadly. We would love to see all of your faces, but unfortunately we can't see you, but we have lots of ways for you to still join in with all the things that we're doing. If you have something that comes up that you think about, either really something that's practical that you want to check, or a question related to something that someone asks that shares today, down at either at the bottom or the top of your screen, depending on the type of device that you're watching this on, you'll see a box called Q&A. In there you can type any question that you like, and as many questions as you like. You're not limited at all, and they come back to us and we can do our best to answer them. So if it's something quick, we can type you an answer. If it's something that we can respond and share with, then we can obviously share that out on screen as well. For those who want to use them, we have some closed captions that are available today. So again, at that bar, either the top or the bottom of your screen, you'll see something that says closed captions, live transcript. If you just click on there, it will bring that up for you. So have lots of fun. Across the next 2.5 hours, we will be finished by 4:30 at the latest. We'll be trying your hand at lots of different things. We will build in some breaks, so obviously you're welcome to come and go as you want to and you need to, but just so you know, if you're keen to do every single thing today, there will be some chances to nip to the loo, nip to the kitchen, and don't have to worry about any of that. We sent ahead a list of some things that you might want to have to hand, things to do some of our crafting, so drawing, pens and pencils. If you've got those, fantastic. Share and join in. If you haven't, we hope you'll still have lots of fun as well. There's lots to take in and watch and listen to and explore together. So have a wonderful afternoon, and I will see you just towards the very end. And I'm going to hand you over now to my colleague Jack, who'll tell you much more about the things we'll do this afternoon.
Hello, everybody. It's brilliant to be on this Zoom call with you all, and thank you very much for the introduction there, Becky. As Becky said, my name is Jack, and I'm going to be giving you an introduction to what we will be doing today and where we are all from, just a tiny little bit as well. But I won't be speaking for very long. I'm going to be handing you over for our first talk and activity soon. So welcome to Curiosity Club. I'll just ask for Ryan to move on a couple of slides here. This is our first time doing a Curiosity Club, so we're very excited. I'm very excited. This is my first time leading a Zoom webinar like this. So we're going to learn lots of things together. Hopefully you will learn lots about the science that we do, and we're going to learn a lot too about how to do these Curiosity Clubs. We have lots of fantastic activities for you. We've got 4 separate sessions, and I'll introduce those in just a moment. Next slide, please, Ryan. That feels very nice to be using that language that lots of us will have seen on television. So who are we? We are a group of scientists from the Wellcome Sanger Institute. And that very fancy sounding institute concentrates on a particular type of science, which is all to do with the tiny weeny codes which are inside all of us. I'm not going to say any more than that now, because our first session is going to be exploring those tiny weeny codes in a little bit more detail. But you can see here on the map where we are based. This is the Wellcome Genome campus. Now, any beady-eyed young people on the webinar today may spot a slightly strange spelling here. Can any of you see it? You may see that that first word isn't the way that we would normally spell welcome. A few people ask us about this. That's because that's the name of the founder of our Funders Institute. And so that is why we have welcome at the start there. The next word there, Sanger, is also a name too. The Wellcome Sanger Institute, named after a very famous scientist whose name was also Sanger, an institute there, the name for our building. So we're just south of Cambridge, as you can see on that map, and a little way between us and London down there in the south of England. Just keep that distance in mind between Cambridge and London, because you may need it for a question later on. But what exactly is it that we're going to be doing today? So again, next slide, please, Ryan. We are going to be talking about two of our research projects, both with quite similar names. So we have the Tree of Life. You'll see the Tree of Life pop up on some of our slides. And whenever you see the Tree of Life, everyone on the webinar today, I would love you to do a little tree kind of movement. I wonder what that might be. Maybe if I were to invent a tree type movement, I might do something like this whenever you see Tree of Life on our slides. So the Tree of Life is exploring those little codes that I mentioned inside all the different living things on Earth. And you can see in our logo there some interesting pictures of different living things. You'll see some more pictures in our second session. The other project that we've got is an extension of this project, I suppose, called the Darwin Tree of Life. So whenever you see the Darwin Tree of Life, I want you to do a sort of bearded man kind of pose. Lots of you on the call will have heard of Darwin, I'm sure, in your school classes. So for that, he's got a nice big beard, some sort of beardy movement, if you see that logo on one of the slides. So you've got a lovely tree for our Tree of Life, exploring the codes inside all living things on Earth. And you've got the Darwin Tree of Life. And as that logo suggests, that's looking at the codes inside the living things that are in the UK and Ireland. So you can see that green shape there, that map of the UK and Ireland. So you'll see those two logos popping up, and they're the two research projects that our scientists are from, who will be talking to you today. But what is it that they're going to be talking to you about? Well, that is on our next slide, I believe. So here is our session plan. I'm not sure why I've titled it in such a boring sounding way there. Our exciting activity plan should be the way that I phrased it instead. We've got 4 activities for you. The first of those is called the Book of Life, and that will be talking about some of those little codes that I've mentioned. We'll have a break after that one, and then we'll be moving on to our second session called Insect in Sketch. That is going to be doing some drawings of some of our flying insects. They're a big focus for our research at the moment. The third session, DN Apple Code, that one is going to be exploring apples, as I'm sure you may have guessed. And if you've got a couple of apples with you, then they'll be very useful for that session. The last one, and I think my favorite named activity today, is Spotty Dotty Squirmer Wormer with Erna. I think if you are sitting in front of your screen at the moment, I'd like you to say that with me, because it's a real delight to say. See how quickly you can say Spotty Dotty Squirmer Wormer with Erna. I'm a huge fan of that title. That'll be our last session. And as you can see there, we've got breaks in between all of those. For each of these sessions, you'll want a piece of paper and a pen if you have it. But as Becky said, if you don't have any of the things that we have, do not worry. You can still enjoy what we have on screen for you. So without much further ado, I think we should get started with our first session. So Ryan, if you go on one, you can see our first session highlighted there, the Book of Life. You're going to have 2 of our scientists talking to you in this session. Everyone, aren't you lucky? You've got Charlotte Wright and Sophie Potter, who I'll be handing over to in just a second. You can use pens and paper here. There'll also be an option to do a little online poll a little later on. And for that, you can use another device if you have one that's connected to the internet. So that might be a smartphone or a tablet or something like that. If you have one of those nearby, that is fantastic. If not, do not worry. You can still enjoy everything that they're doing without it. So without saying very much more at all, I'm going to hand over to Sophie and Charlotte. I think if we go on to the next slide, Ryan, and I think you may be able to see 2 of our logos on there. So I'm going to do it and pass you right over to Sophie and Charlotte for the Book of Life.
Thanks, Jack. Thanks. So yeah, today we're going to be talking to you about the Book of Life. But first, who are we? Like Jack has just said, I'm Charlotte and I'm a scientist at the Sanger Tree of Life.
And I'm Sophie and I'm also a scientist supporting the Tree of Life programme.
Yeah, and so just as a quick recap, that today you're going to need a piece of paper per person if you'd like to join in, some coloured pens or pencils, ideally 4, and a second device of internet. And we're going to be talking about something that every living organism, which is also known as a living thing, has in common. So first of all, we thought we'd get you thinking about differences first. So here we have 4 living things that you probably recognise. We've got the apple tree on the left, we've got a mushroom next to that, a cat all squished up in a box. Maybe you've got a cat at home that likes to do this. And on the right, a bumblebee. And so we first thought that we'd get you to talk to somebody in the room if you've got family members with you, or if not, maybe just think in your head or write down differences that you notice between either, say, 2 of these things, like the tree and the mushroom, or the mushroom and the cat. What do they have and what's different with them? And as kind of a real challenge, if you can think of anything that is different about all 4 of them. And we'll give you just 1 minute to think about that. So look at the pictures and think about what's different. And Sophie's also going to have a think as well.
I'm going to try and decide what I think is different about all these things. Well,
what are we thinking here? We've got a nice big tree. That's quite small.
I mean, 2 of them have legs and 2 of them don't, but that's not different between all of them.
Cats, isn't it? The eyes are different size.
There's only eyes for the bee and the cat. Yeah, I think there's definitely things that some of them have in common and some of them don't. I mean, they're all basically different things, but there are definitely some similarities going on too, I guess.
Yeah, so I guess we can start talking about things that you might have said at home. I wonder if any of you said, well, a tree is a plant, whereas some of the other things like the cat is an animal. So there are different types of living things. Maybe some of you said fur, because the bumblebee looks quite furry and so is the cat. But they all have quite a lot of differences between them. Some move, like the cat and the bee, whereas you don't really see plants and mushrooms moving, do you? So there's quite a few differerences between them, and I'm sure a lot of you at home have got many of these things and more things that perhaps we haven't spotted. But they also have, things in common. And so again, if you maybe think with somebody at home or write down anything that you can think of either 2 of them have in common, like the cat and the bee, or things that all of them have in common. And this one's probably the tricky one. What do they all have in common? And again, just maybe think of somebody at home for a minute.
Well, like you said, there's some furry ones, some with legs and some without. I would personally only eat the apple and the mushroom and not the other 2, but some people may vary.
True.
Yeah, what do they all have in common? Well, they're all alive, obviously.
True. Yeah, and I think a lot of people maybe say that at home. That's a good one. They're all alive.
And they all grow and they all have babies in their own way.
Yeah, that's true as well. They all grow. That's an interesting one. We'll wait a little bit longer. See if people are thinking. But yeah, so there's lots of things common between two of them or between three of them. But what they all have in common, other than that they're living and they're alive, is that they grow. So the apple tree starts from a seed and it develops into the huge trees that you see. Equally, a cat, you may know this starts a kitten and they grow a little bigger. And a bumblebee once was just a tiny little egg that then was a larva that grew into the adult form. So every living thing grows and develops, but they end up looking very different. But so how do they know how to grow? And the answer to that maybe some of you said, if you've heard this word before, is the word genome. So every living thing has this book inside them, which we call a genome, and that contains the instructions for for them to grow into the living thing that we see, and what to end up making them be, to look like. So if we have a look here, the apple tree has a genome, and that genome will tell it to grow apples and to have these nice big leaves that absorb light. And then the mushroom has a genome that tells it that the nice bit at the top is going to be red with these little white dots. The cat has one, presumably somewhere it says squeeze yourself inside a box, but also have fur, but don't have fur that's all the same color, have some bits that are orange, some bits that are white. And the same with the bumblebee. Somewhere in its genome, it's telling it to have wings and things like this. So these set of instructions that we call genome, and each living thing has one. And the genome is made up, the genome is made up of codes. So if we look inside these books, you see a sequence of letters inside each of them, and the sequence of letters is different for each of them. If we quickly zoom in on just a few of those letters in each of those codes, you may notice a few things. Again, they have some things in common and they have some things that are different. So I'll give you 1 minute to have a look at these codes, the code for the apple tree, the code for the mushroom, the cat and the bee, and to have a little think about what do you notice that's in common between the 4 codes and what is perhaps different between the 4 codes. And just think again of someone at home, or write it down, and Sophie's having another big think.
Having another ponder here.
We're making you think a lot today, aren't we?
I know, it's a Saturday as well. Yes, so obviously this isn't the full code, but it's a section of the code. Yeah, you can see it's made up of letters, and it looks like the letters are pretty much similar, but they're in different orders and in different places depending on what type of living thing we're looking at.
Yeah, so we'll give people at home just a little bit longer, but maybe they're already noticing some similar things.
I think so. Yeah, I'm definitely seeing, there's not the full alphabet there, that's for sure.
No, exactly. So if we come back together now, yeah, exactly. There are only 4 letters that you're seeing here: A's, T's, G's, and C's. Like you never see a Z, you never see a U. So it's always the same 4 letters. But they're in different orders, like you say as well, in the genome. So, for example, in the apple tree, it starts with an A, whereas in the mushroom, it starts with a T. So the same 4 letters, but different combinations. And in fact, these long codes that we've been talking about, they have a fancy word that scientists like to use, and that word is DNA. And you may have heard it before. But I wonder how many of you know what it stands for. It actually stands for this word, Deoxyribonucleic Acid. And that's quite a fun word to say,
it is.
I would say. So maybe we say it one more time, and then Sophie, you can say it. And when Sophie says it, you can say it with her at home and see if you can say it. So deoxyribonucleic acid.
So deoxyribonucleic acid.
Cool, it's a cool word. But yes, it stands for DNA, and that's generally what we will refer to it as. And so just showing you here, some of you at home may recognize this shape that DNA makes. Maybe have a quick chat with someone in the room if you recognize this. If it's fine if you don't, but some of you may recognize it as a specific shape that DNA is. So some of you might be saying the word helix, maybe double, because it's the 2 strands. And yeah, exactly. So it's a double helix. It's just a fancy word that we call DNA. But if we take a look at it, you see that it's these 4 letters. So the code is made up of the 4 letters, G, T, A, and C. And you'll also notice that we're drawing the G's in a color, yellow, T's in green, reds in A's in red, and blues in C. And the letters aren't really actually these colors, but we use it to make it easier to look at and for us to see when we're studying DNA. So we give them different colors. So that's a lot of us talking and explaining things. So I think it's about time that you did something at home. And in fact, we're going to draw a code. So we've been looking at them, we've explained what it is, and now we're going to draw one. So for this, you're going to need a bit of A4 paper, and Sophie's going to do it alongside you. And once you've got that bit of A4 paper, and don't worry if you don't, but if you want to join along, please do. So fold it in half once. And again, once you've folded that.
Testing my foldings.
A lot of folding. One more time.
One more time.
That's now 3 times you've done it. And can you fit one more fold in?
Yeah. We can put down for that.
This is extreme folding.
Extreme folding. This is origami on a.
So you've got a total of 4 folds. And if you now space out the bit of paper again, how many sections are you counting there?
16. So
16.
One circle per section.
Yeah.
Right, so you should be able to see that each fold is has made a section. So we've got 16 sections in this piece of paper now.
Exactly. So we'll give you another little bit at home. It's time to do that. And then if you're already speedy and you've already made your 16 sections, then I'd ask you to draw a circle per section. So you've got the folds to make those circles nicely, evenly spaced. Do a circle per section. Sophie's going to show you what we mean shortly.
So it's going to look something like this with the circles. I'm going to keep drawing.
And hopefully you're able to do this at home too.
Oh, drawing a straight
Nice
line.
big circles. If you only manage to do 3 folds, if the 4th fold is a bit too tricky, that's fine. That just means then you do 2 circles per fold. But if you've done the 4 folds, you can do just 1 circle.
So when you're finished, you should have a piece of paper that just has a load of circles running along it like that.
Excellent. So you have plenty of time more still to do this, but for people that are speedy, I'm going to now ask you to do the next step, which is there are 2 codes at the bottom of the screen, both starting with blue. And I want you at home to pick one of those 2 codes and use the key at the top of the screen to draw the corresponding letter in each of your circles in your paper. So for example, in the first circle, it's blue, and blue is a C in our code. So Sophie would draw a C as her first circle on the page. And if you have colors, you can do the C in the actual color blue. So you're drawing a blue C. But if you just have a pen, then you can just do it all in the black.
I'm just using it in one color, so all of mine are going to be blue, but I will write all the different letters out.
Yeah. And so as you're just doing this at home, we'll chat a little bit more. So this, what you're actually doing is part of the human genome. This is a code that's part of it. Whether you pick the top one or the bottom one, it doesn't matter. Maybe if there's 2 of you or more, pick different ones to make it a bit more fun. But this is a real sequence or code from the human genome that's inside every one of us.
I have a code.
Awesome. Do you want to show it?
Yeah.
We've got an excellent code here. And we'll be able to check at the end if you have managed to work out the correct letters for each colour. And we'll show you the answers at the end. We've also a bit of a surprise as well that Sophie's got for you.
Also might tell you what your code actually means.
Mm-hmm.
So I'll just give this another minute just to finish off, and then we've got a little poll to ask you as well in a second.
Yeah. So if you're struggling a little bit, so we've said that the blue is a C, so the first circle would have a C in it, and the second circle is red, and red in our little key at the top is an A, so you draw an A in it. That was very quick, that's great.
Had to struggle for a minute to remember my colours, which I wasn't doing very well. It's quite difficult with the 16. You get halfway through and forget where you are in the code. I hope everyone else is not finding it quite as difficult as I am today.
Okay, so don't worry if you haven't finished it, but if you have, or however far you've got to, keep it somewhere safe because we're going to come back to it at the end. But hopefully you've managed to do a bit of that. So we've just drawn 16 letters from the human genome, but the human genome is a lot bigger than just those 16 letters. We've just drawn a small part of it. So my question for you guys is, if you were to keep drawing every letter of the human genome and they were still these equal spaces apart, how big would your bit of paper need to be? How far would that code go if you kept on drawing those circles? Would it go for the whole genome, that is? So would it go from Cambridge to London, which you may have seen on the map earlier that Jack showed you? Would it take you from London to Brazil? Would you go all the way around the world, or would you go to the moon and back? So have a quick think about that, and we're going to have a poll up that you're going to be able to answer. So how far would your genome go if you were to keep drawing it?
So a poll should have just appeared on your screen. Unfortunately, it says panelists can't vote, so we don't get a vote on that.
Yeah. You can whisper it to me what you think.
Like a secret. But yeah, so is it as far as London from Cambridge? Is it as far as Brazil? Would it go all the way around the world, or would it go all the way to the moon and back?
It's quite a tricky question. So you guys got to think, how big is it and how far would that take you?
I still struggle to imagine how far it is to go all the way to the moon. It seems like a very long way.
Brazil is also quite far away.
It is, that's true.
And if you take the train, it can take quite a long time even to get to London from Cambridge.
That is true.
We'll give you a little bit longer. We'll give you 15 more seconds at home, and then we'll reveal the answer and see if you got it right or not, or how close were you to the right answer.
Yeah, it is. I have to admit, before we started writing this exercise, I didn't even know how far it was. So it was.
So the scientists have been learning things too.
I've definitely learned things doing this. Okay.
And the answer is it going to appear? Yes. Whoa. Ooh. So 13% of people said from Cambridge to London, 7% Cambridge to Brazil. Around the world was very popular with 47%, and all the way to the moon and back was 33%. Sophie, would you like to reveal?
I will, yes. So in actual fact, the majority of you were correct. It would stretch all the way around the world on that scale, which is an awful lot of letters to imagine.
Yeah,
and quite a bit of pieces of paper. So yeah, it's a massively long code that tells us how to be who we are.
Yeah, and in fact, if you were to count each of those small circles, you'd be counting 3,200 million times. A million itself is quite a big number, let alone 3,200 lots of it. So this is a huge code.
I'm fairly sure I would lose track of counting quite early on in that process.
We're not going to make you write them all up today, luckily. But we are now going to talk to you about other living things that have genomes, and I'll pass you on to Sophie to tell you a bit more about that.
Yes, so as we mentioned, the code that tells humans how to be humans would stretch all the way around the world, but different living things have obviously different codes to tell them how to be themselves, and some of these codes are much shorter than others and some of them are longer. So what I want to do is just take you through a couple of different living things and thinking about how big the instruction book that tells them how to be them would be. So the first thing I want to think about is a grasshopper. Now on this screen, the grasshopper's looking pretty chunky, but I think most of you have probably seen grasshoppers before. They're generally sort of this big, and you see them in gardens and in fields and a lot in the summertime. So it's a sort of small insect. And I wonder if you think, would the code that tells a grasshopper to be a grasshopper be larger or smaller than the code that tells a human how to be a human? And I'll just give you all a couple of seconds just to think about that. Maybe tell people you're with what you think. Charlotte, do you have a feeling about it?
Well, I feel like it's small, so maybe you would expect it to be small, but I feel like you might be catching us out. I'm not sure.
This might be a trick question. Well, That's a very good impulse that you've got there. Amazingly, the code that tells a grasshopper how to be a grasshopper is actually almost twice the size of the human code. So if you were to write that out in the same way, it would stretch around the world almost twice. So that's really quite amazing when you think about it. The next living thing I want us to think about for a second is the pumpkin. And I imagine people are seeing quite a lot of pumpkins at the moment. Maybe you've even gone to a pumpkin patch recently. One of my friends is there today. And they can grow really massive. I mean, if you look at the size of this pumpkin compared to the people looking at it, it's really a huge thing. But I wonder, do you think the code that tells a pumpkin how to be a pumpkin is bigger or smaller than a human code? Talk to the people that you're with, see what you think. You know, it can grow very large. So maybe we think it's got a big code. On the other hand, I've been tricky once before, so perhaps that's what's happening here too. Well, what I've got to tell you is, actual fact, the code that tells a pumpkin how to be a pumpkin is only about 1/10 the size of a human's code. It's really quite small. And if you were to write that out, you would only get as far as the very eastern tip of Canada, which is still quite a long way, but it's definitely not all the way around the world. But the one thing I want to make sure you don't think is that plants in general have shorter codes than animals, because that's not actually true. So far in our project, the longest code that we've worked with has actually belonged to a plant. It's belonged to mistletoe, which is a plant you sort of associate with Christmas a lot more. And the mistletoe genome is almost 10 times as long as the human code. So it's really quite large. It's quite impressive. So we've got one more activity for you now. And as we've said, living things come in lots of different shapes and sizes, and every single living thing has a genome, has a code that tells it how to be itself. And so we want you to think a bit more about the different types of living thing. And so we've got a little poll for you, and we just wanted to go to this address here and type in this code, and all you need to do is name as many living things as you can think of that's names begin with a D, an N, or an A. And once you put those in, you click select. Hopefully in a minute, we'll be able to see all of your suggestions and see what you think. So if you have a device, if you can log onto that site now and type in that code, and we'll also have a think about things that begin with D, N, and A, and just give you a couple of minutes to do that.
Yeah, and I guess maybe think how different those things are. Can you think of things that are really different from each other that begin with D, N, and A to make it even harder?
It can be animals, it can be plants, it could be anything you think of, really. It can be microbes, if you can think of something like that.
Tiny little things.
We know that we have some of our friends in the audience who probably are thinking of these things. And we'll just give you a couple of minutes to think about that.
I can think of something at home. Maybe some people have that start with a D.
Oh, yeah.
Something quite friendly. And soon we should be able to see the results of what you're thinking of and what animals or plants you've come up with, starting with these letters.
I'm thinking of things people like to swim with sometimes as well. Yeah, there's lots of different things.
What's the funnest thing you can think of?
The funnest thing, well, I have a personal favourite actually. It begins with A, and it's called an axolotl that some people might have seen in pet shops before. It's a really cool little amphibian type thing, and it has these really great gills that show up, and they always look really happy. And that's probably my favourite.
I don't know if my thing counts, because I was going to say a dinosaur, and it is a type of living thing, but is it still living?
Well, it was a living thing. I mean, some of our friends also said dodo before, so that's definitely something we can have. Let's click through and see if we can see the Menti page. Oh, there we go. Click S.
Oh, wow.
Yes, we've got something.
There's some excellent dodo. Use dodo.
Dodo was a possibility. Ant, yes. Narcissus, that's a type of flower, isn't it? Oh, look at all these options coming in.
Wow. Dark, yeah.
Alligator.
Apple tree. We're going to be learning some more about apples later.
Yeah. Also damselfly and dragonfly. Yeah.
Those are good ones.
Anteater. Oh, really wish we had anteaters here. Right, these are great suggestions. Thank you much, everyone. I think we're going to have to move on because I've just noticed what the time is. But these are really great. Thank you so much for giving us all your suggestions. They're brilliant. These are some of the things that we came up with when we were brainstorming all the different things. Some of them are things that you've probably seen quite a lot, like nettles and ants. Some of them are a bit more exotic, like the dung beetle and the alligator, and of course the dinosaur that you mentioned earlier. There's some spoilers here for things that are going to be happening in the next session. So you'll notice that for D, we've got a lovely damselfly, which is one of the things that Petra will be talking about. There's the A for apple, which is what Max will be speaking about. And then this cool little worm thing that you can see next to the N, that's called a nematode, and that's something that Erna's going to be introducing us all to later. So just to round us off, just wanted to show you this place again. This is where we work, the Sanger Institute. It's where the human genome was first studied, and it's where we are now working to study a whole lot of other living things. We want to find out what makes their codes different and how that makes the living thing itself different from each other. We really hope that when we're able to do in-person activities again, you'll all come and visit us, because we've got a new and longer version of this called the Long and Winding Code, which will be a walk through a genome that we'd love to do with you. And finally, the code that you wrote down earlier, I'm sure you'll want to know what that means. So there were two different codes, and there's one letter different between the two codes. And depending on which code you wrote down, if you wrote down the top one where there was two G's, that means that you have wet earwax. If you wrote down the bottom one, which is an A and a G, you have dry earwax. So you might want to check whether the code that you wrote down actually matches your own type. But with that, thank you all so much for being with us, and we'd like to pass you back over to Jack now.
Hello, everybody. Thank you very much, Sophie and Charlotte, for that fantastic introduction to DNA and our genomes. I hope that everyone on the call had some fun doing that. It's fascinating, isn't it, that these tiny little codes inside us that have all these letters in. I wrote that down, 3,200 million letters in the DNA code, the genome that is inside you and that's inside me, that gives our body the instruction of how to grow and how to live. And I don't know about you, I did that exercise. I wrote down this one, which means I have dry earwax if I have this part of my DNA code. I wonder if I check that. Oh, actually, I'm not sure if that is right. Maybe it should be the GG wet earwax version. I don't know. How about you at home? Have you got wet earwax or dry earwax? If you have a check, then you can see this code on screen is going to be part of your real DNA code that's inside of you. I'm going to ask Ryan if you can move forward on the slides, just in case anyone's wondering. Ryan, by the way, who I keep mentioning, is our fantastic technician on this call. I don't have control of the slide deck, so he has to move them on for me. I'm not just doing an impression of those COVID meetings that we've seen on TV. Oh, I can see, thank you all very much for doing that first session, the Book of Life. We are going to be going on a break shortly. Just before we do, on the next slide, you'll be able to see what our next activity is going to be. So after we come back from the break, I'm going to suggest we have a 5-minute break. So according to my clock on my computer, it's 2:41 at the moment. So if we come back at 2:46, just after 2:45, then we will be starting our second session, which is Insect in Sketch with Petra, another one of our scientists. For this session, again, you'll want some paper if you're going to join in with us, and the minimum requirement you might need is a pen. Ideally, the things that you'll see Petra using: pencil, rubber, and some colours as well. But as we've said all the way through so far, if you don't have any of these, then you can just spend a lovely time learning with Petra in the next session. But we'll go on a short break now, and I'll see you all back here in 5 minutes' time. All right, bye. Slide. Just before we get going with Petra, I'm going to introduce a little more about what we do at the Wellcome Sanger Institute, and as part of our Tree of Life and Darwin Tree of Life and Darwin Tree of Life research projects. So as you heard in Sophie and Charlotte's session, we're looking at DNA, at those genomes, that full DNA code of different living things. But of course, in order to study that DNA code, we first need to collect some of those life forms for us to study. And to do this, our scientists go out all over the UK and Ireland to collect these life forms for us. And we've got a little video of them doing just that. So Ryan, if we go on to the next slide. Right, so that was some of the techniques that our scientists use in the field to collect the samples that we're going to be looking at the DNA codes of. And most of those things that our scientists were collecting there, a place up in Scotland called Benai, up in the sort of middle of Scotland, were insects, you might have noticed. And that is useful for us for our next session, which is all about insects with one of the insect team. So for this next session titled Insect in Sketch, I'm going to be passing you over to Petra. So as this is a drawing activity, we wanted to have a couple of different cameras on Petra. So you will notice that this part of our session today was actually pre-recorded. So Petra won't be able to respond in the video to any questions that you put in our Q&A box, but she is on the line with us on the call. We're very excited to have her with us. And so if you do have any questions as we're going along, please do put them in that Q&A box for us. And also, as you will hopefully be doing some drawing with us, we'll share an email address at the end to send in pictures of what you create. We'll be very excited to see that. So I will move us on to the next couple of slides, actually, I think. Yeah.
So hello, everyone. My name is Petra, and I am a scientist here at the Wellcome Genome campus, working on malaria mosquitoes. So in my free time, and sometimes in my work time, I like to draw a lot. I've always liked to bring my art side into my science side, because I found that art really helps to convey my science better. And it also makes it way more entertaining. And not just scientists, but also people outside really enjoy seeing science represented in that way. So what I want to do with you today is have a little art class and a little bit of a biology class, and teach you about the anatomically correct way to draw insects. So for this activity, you will need any kind of paper, eraser, pencil, some colored pens, pencils, whatever you have at hand, and a marker. In this case, I'm using a black marker. Because my art supplies are all water soluble, so they basically smudge if in contact with water, I will be doing pencil sketch, then coloring, and then adding lines. You can follow that way, or if you prefer, you can also switch and then do first the lines and then the coloring. If you have pencil, that's probably going to be easier. So what we will start with first is we will talk a little bit about the anatomy of insects. So what is this word anatomy? This is basically how different body parts in living things connect to each other. So if we divide the paper in about 1/3, 2/3, on this side, think a little bit about how you would draw a person. So the anatomy of a person. And as you're thinking about it and drawing it yourself, I'll draw it how I would usually do it in stick figure form. So we have a head, the neck, the body, and then to the body we have 2 legs. This doesn't need to be perfect, and 2 heads. We can also draw a little smiley face. So if I try and describe the anatomy then of this person, we have a head. So this is the head. Then we have the torso, which is divided into 2 bits. The upper part, which approximately ends at your waist, is called the thorax. And your hands attach to the thorax. And then the bottom part from your waist down is called the abdomen. But they do kind of blend together, so this is not a harsh line. So this part is the abdomen. And so your legs attach to the bottom of the abdomen. And now that we have colored this in, I'll add just the outlines to make sure that they're nicely visible. So we have the lines. It doesn't matter if you go outside or inside the lines. That's your artistic freedom. So we have a head with all of our facial features. We have the thorax abdomen part in the middle. And we have the hands and the legs. In total, humans have 4 limbs. Okay, so now we have the anatomy of a human. So what does the anatomy of an insect compare to these parts? So like I said, I work on mosquitoes. So let's draw a mosquito next. And you can think a little bit about how you would draw a mosquito or just follow me. So the mosquito has the same 3 parts, except these 2 are divided. So you can draw a mosquito by drawing a circle for a head, an ellipsis or a squished circle for the thorax, and another squished circle, long one, for the abdomen. So it has exactly the same 3 body parts. And again, on the head, like humans, mosquitoes have facial features, so in this case, a really long nose with which they bite you and drink your blood, 2 eyes, really big eyes, these are kind of small, and 2 antennae. Now, unlike us, where we have our arms on the thorax and our legs on the abdomen, mosquitoes have all of their limbs on the thorax part. That includes 6 legs. So 1, 2, 3 on this side, and then just a sign of 1 on the other side. And they also have joints like we do. So you can do 1 joint, 2 joints, 3. There's more, but for simplicity's sake, we will keep it as 3 joints. And just do these elongated shapes. And then on the other side, there's another 3 limbs. So mosquitoes have 6 legs. And I say legs because insects also have wings. Mosquitoes are flies, so flies have only 2 wings, while most other insects will have 4 wings. I say most because you will see later there are some cases where it's slightly different. So wings, flies have 2, and other insects have 4. So now that we have, oh yeah, and again, 2 antennae. I may or may not be spelling that one wrong. Okay, so let's color this in. So you have a visual comparison between our little stick figure here and our little stick mosquito here. So again, we have the head, then the thorax part is quite big. It includes the thorax, it includes all 6 legs, and the wings. Mine is going pretty fast because markers are really practical at coloring really large portions at the same time. However, you can see some smudging. So this is not the greatest paper for markers, but we will make do. And the abdomen part, which has no limbs attached to it. And if the mosquito has drank quite a bit of your blood, this is gonna be full and red. Okay, let's add some details. Let's just make sure that this is dry because I don't want the marker smudging. So, big nose. Huh, that part is a bit off, but that's fine. 2 big eyes, 2 antennae, 2 wings, the thorax part, and then 6 legs. I will leave the 3 legs behind without any lines, so it seems like perspective-wise, they're behind, and it's a bit easier to see. So 3 legs on each side. And we have the abdomen, which usually has separate parts divided by lines. So this is our anatomy bit. As you can see, humans and insects have quite similar parts, with the 3 main one being differently shaped, but named the same, and they do the same purpose. So what about, while you look at this picture and what I mentioned here, what about other insects? So flies are just one group of insects, and insects are some of the most varied organisms, living things on the planet. They look very different from each other, even within the same group. So let's work on some other insects. One that is very popular, very well known. I don't know which insect popped into your head when I mentioned that, but let's talk a little bit about the anatomy of butterflies. So butterflies are some of the most commonly drawn insects. Everyone likes to draw butterflies. And you don't need to draw the next few things because I'm just quickly going to sketch. The way people mostly draw butterflies is they will have a really fat caterpillar, maybe they'll add some antennae to it, and sometimes 4 wings, but I've seen people draw butterflies with only 2 giant wings. So, as you have learned in the first drawing we made, this is not quite a correct adult insect. So the middle part, like I said, is a caterpillar. And caterpillars are baby insects. A baby fly is called a maggot, and they're this little wormy shaped forms of insects. Basically, if you can imagine, this is the baby insect. And the only job they have is to eat as much as they can so they can grow into adults. And they transform into adult insects. So once they transform, they now have the same 3 parts that we talked about. They're not wormy shaped anymore. So if we were to now draw a butterfly anatomically correct, we need to start with the same 3 pieces. So the head, the thorax, this is going to be a butterfly from above. So similar to our wormy butterfly over here. And a really nice long abdomen. So the mouth parts of a butterfly are different than a mosquito. They don't bite people, they just drink plant juices. They still have 2 antennae, and they still have 2 large eyes, but their mouthpieces is usually just this really nice long tongue that they use to suck plant juice. And again, all of the limbs, so all 6 legs and 4 wings, attached to the thorax. So I'm gonna make the wings slightly smaller so they fit on the page, but butterflies can have really big wings compared to their body size. And the ones that have a really big fat body with small wings are moths. So let's draw. The wings very often also overlap, so don't worry about the lines. You can erase them later on. And of course, wings have really beautiful patterns very often, and I encourage you to go and just look at pictures online of various different patterns on butterfly wings and see if you can recreate them in your drawings. So let's color this in with the same colors we used before. So again, this butterfly has a head part, and because the wings are so big, we don't see the small legs below it, so we will draw this on the side in a bit. Then there's huge wings. I'll try and quickly color them in. Don't worry if it's not perfect. We are still just in the learning phase. I might need a new green marker after we're done with this course. I do very much love green, so I use green a lot. Okay, so that's our thorax parts, and then we get to our abdomen. So let's wait for that to dry a little bit, and let's start adding our lines. So we have really big eyes. And one thing I didn't mention, but you can look at insect faces, and the eyes are what we call compound eyes. So the eye of an insect is actually made out of a lot of small eyes all together next to each other. So have a look and see if you can spot how many eyes. Don't try and count them. There's a lot of eyes in there. Then we have our little mouth part. Okay, and then thorax. And abdomen. Okay, so these are the 3 bits. Now if we look at the same butterfly but on the side, similar to how we drew the mosquito, that would be head, thorax, abdomen, really big wings. This one goes behind here, the antennae, mouth part, eyes, and then butterflies usually have very small legs. They don't need to use them for much. They're not scuttlebugs, they don't run around, they just mostly fly. So again, teeny tiny legs, 3 on each side. Let's just hint at the 3 on the other side. So that's our butterfly from the side. And again, let's just quickly color those in so you have the colors to look at and compare the 2 views. And the abdomen. Okay, so this is our little side view butterfly. These are usually thinner, but that's going to be harder to see if we draw them too thin. again, we're not going to color in the legs that are behind, just for clarity. And then abdomen. So these were butterflies. Butterflies are very often some of the most drawn insects just because they're really pretty to look at, and people really like butterflies. But what about some other insects? So, can you recognize what this is? So this is a dragonfly, and similar to butterflies, they have a really long abdomen and really long wings. Their wings are often see-through, so you can actually see the legs below, but they have a similar principle as butterflies. 4 long wings, 6 limbs, really long abdomen. So what about some very, very common, but also somewhat odd anatomically looking insects? So can you recognize this insect? So this is a beetle, and beetles look very, very different on the outside. When you look at them, you only see this really hard shell. And only once they open up their wings do you realize that all of the anatomy is there. It's just very well hidden. So they do have a head, which can be very oddly shaped. And then their thorax part, especially their wings, they have completely differently shaped wings. The first pair is really hard shell wings that close off the body, so it looks like the whole beetle is just the thorax. But then when the beetle takes flight, it opens up the outer wings and unfolds these really long see-through wings, and then it can fly off, and behind all of that you can now see the abdomen. And so beetles can be very complicated and they look very strange. Sometimes it's really difficult to see the 3 different pieces. So just have a look and see if you can distinguish those. So that's beetles. Let's go for an even odder one, but still a very common one. So I'm pretty sure everyone can recognize this insect. So this is an ant. And what everyone knows about ants is you see them scuttle about. You don't see them flying about most of the time. So only the queen and the king of ants have wings. And you've probably seen some of them during some times of the year flying around. Once they establish a new home, they lose their wings and basically just have the 6 limbs. And all of the ant workers and ant soldiers don't have wings. So they will only have the 6 limbs and, as I said, scuttle about. So you can see that there's really a lot of different shapes and sizes, and they're really interesting to look at. So now we have done the anatomically correct part. And what I want to do next with you is we will take that knowledge and draw one of my favorite things ever, which is a very sassy mosquito. So I like to draw my mosquitoes with a lot of expressions, a lot of human expressions, as if they were in a comic book. So in this part, I want you all to follow along and see how your sassy mosquitoes are going to turn around. This is going to be a bit slower because when I draw, I like to make sure that all the shapes are in the right position, and we will make sure that we add some more details at the end than we did previously. So we start with the basic 3 shapes, but we tilt them slightly because I want my mosquito now to walk on her hind legs. So we start with the head, and now you can see I'm going around the lines a few times. We will erase this lightly in the end. So don't worry if your lines are messy at this point. So we have the head, then we have the thorax and the abdomen. Make your mosquitoes as fat as you want. Okay, so we have the basic body shape. Now let's add a big nose and 2 eyes. And this is where I start taking out my artistic freedom, and we will add a pair of sassy eyebrows. Mosquitoes, like other insects, don't have eyebrows, but we can cheat a little bit and make her look very suspicious. And then we can add 2 antennae on the top. And so what do we do with the limbs? I like to put the limbs in odd positions where normally the limbs of an insect would never be. But again, we do have some artistic freedom now. So I'm just gonna sketch 3 bits where I want my limbs to go, and on the other side. And now I'm gonna just line up, and I want her to have a pose like this. So let's do that. We will add 2 upper arms. So I want her to have 4 upper arms in total that are very judgmental. And then I'm gonna use the other 2 limbs as really long legs. And I also want them in this triangular shape. Like she's standing like this. And now that we have our lines, you can just go in and add these curved lines to make the limbs have a bit more shape. And this side as well. And then we can add hands as just half moons. This one's behind. Don't worry, we will erase these lines before we start coloring. So then the legs. Again, just curved lines to add a bit more meat to the legs, and some half moons for the feet. Okay. So what we are still missing here is the 2 wings. And we're gonna add 1 on each side. Again, they attach to the thorax part, so let's give her 1 elongated here. Just some curved lines. And 1 on the other side, like she's about to take flight. Okay, and so you can also just add some approximate lines on the abdomen. So at this point, you can have a look at your sketch, see if you are happy where everything is. And since we want this to be our final drawing, what you do now is you take your eraser and just gently press it to the paper. So we just want to remove some of those extra lines. I'm not gonna go over the eyes because I do like how they are placed, but the leg lines that we used just to see where everything goes, all of the double lines. So I recommend soft, or in my case, a softy eraser for that. That doesn't smudge too much. And also if you're using so I'm using a 2B pencil. You can use an HB pencil or an H pencil. They make much fainter lines and are easier to erase. Okay. Okay, so once you're happy with the lines and you've cleaned this up a little bit, now let's go in with the color. Again, at this point you can also go in with your black pencil, but because, as I said, my black pencil and my pens are all dissolvable in water, so I need to go first with the color, otherwise the drawing is all going to be smudged. And now we're just gonna color it however we want. So I don't want to color the head and the thorax and the abdomen differently. I just want my mosquito to be very orange. And so let's go in and let's color her orange. So the idea is I still see where all my shapes are because I didn't erase them so much, but they're not gonna be visible once or they're gonna be less visible once I actually add the black lines. Okay. You are more than welcome to have more colors and use more colors when you're doing this. I unfortunately only brought the 3 today, so. But what I want to do now is, because these are watercolor markers, just add a little bit more color to it because they can be layered. So now I can add a little bit of a shadow here and there by just going over some areas again. You can see that looks more orangey. Again, if you have pencils, you can use a differently colored pencil at this point. It's probably going to be a bit easier. This is just paper I pulled out of a printer, so it's not the best paper to draw on, that's for sure. So yeah, just adds a little bit of extra color here and there. Okay, now let's go to the wings. Oh, it's okay. We can fix some of the smudging with the lines later. Okay. All right. So that's our base color. Now I really want to be sure that that is dry before we go in with the ink. Okay. I'm gonna be a bit slower now because I want my lines to be a bit more stable. Don't worry if you can't do a line in one go. Just do it in a few times. Like, I can't draw a circle from just one go, so I'll just draw several tiny bits. Now this is the chest part, so all of the limbs on that side are behind. Oh, see, I do have a little bit of smudging. I should have waited a bit longer. That's okay. So let's add limbs. Goes behind the leg. Don't need to do this in one go, especially for long lines. Just go a few times. Okay. So abdomens usually have lines like this, but what else you can do is, if you look at mosquitoes under a microscope, they are covered in tiny scales that look a little bit like fish scales. So you can add a little bit of detail by just adding a few little scales here and there and around the thorax part. Okay, let's just finish with the wings. So the wings do have a lot of detail on themselves. You can look at photos of mosquito wings and see how very intricate they can be. I usually just draw one curved line and then a few curved lines inside there. So it gives the impression of a wing, but you don't go crazy trying to draw all of the details. And a few curved lines. And that's it. And you have now successfully made a drawing of a sassy mosquito. So in this course today, I hope what I've conveyed to you is that insects are very interesting, have very interesting shapes, but at the end of the day, there are some rules you can follow to make your drawings a bit easier. So what I encourage you to do now is to just go online, look for really interesting insect photos, and try and recognize the shapes, try and recognize the various different bits. Some are going to be really complicated, that are even complicated for me, where I'm not sure what's the head, what's the thorax, what's the abdomen. And yeah, just try and draw some of them. Draw them a bit more seriously, or draw them in a completely comic form. This one gives you way more artistic liberty, so I would go with this one. That's the one I also enjoy drawing. And yeah, see what other crawlies you find, because what I've been talking to you about today were insects. However, when you search around online, very often you will have some other animals in there, like spiders or mites. scorpions, all of those are not insects. So their anatomy is going to be very, very different. And try and see how it differs, which parts connect or merge together, how many limbs they have. You probably can guess just from the name that a centipede is going to have way more legs than an insect. And with that, I hope you had fun with this drawing class. I hope you go forward and make many interesting insect drawings. And that's it from me today. Goodbye.
Hello, everybody, again. What a fantastic session there with Petra. I wonder what your sassy mosquitoes look like. I've got mine here. I put some different colors in there. I'd love to see all of your different drawings that you've done. We've got our email address on there for you now, engage@welcome. Remember that unusual spelling of welcome, welcomeconnectingscience.org. Please do send those in. We'd absolutely love to see them. And I think Becky can put that email address in the chat for everyone as well, if it's a bit easier. We are going to take a slightly longer break now. So we'll take a 10-minute break. But when we do come back, yeah, I can see Becky has put that address in there for you. When we do come back, our next session is going to be our DN Apple code activity. For those, if you've got them, do bring along your 2 different apple varieties. I've got something here that's lovely and green, slightly bruised actually. I must have dropped it somewhere. And this one, which is a lovely russet color. You're going to hear what that russet color means. We also have a sheet for that activity that you can print out if you have a printer there. Becky is going to be putting that Word document in the chat for everyone too. So if you have a printer, you can print that out. Don't look at what's on it though. Save that for when Max gets to it in his session, which is just up next. But if you don't have a printer, it's absolutely not a problem. You don't need to print that sheet off. You can do the activity just as well without it. So we'll be going off. Yeah, Becky's put that in the chat now. Fantastic. We'll be going off on our break now. We'll have a 10-minute break, so coming back at 1:45. Just to make sure that we keep in time, we won't do a break in between sessions 3 and 4. So this is a good point to go and do any of those things that you need to do before we finish at 4:30. Yeah, back together in 10 minutes, everyone. Looking forward to seeing you all there. Hello, everybody. I hope you had a brilliant break. We are very excited to go on into our next sessions. And we've been very excited too to receive our first drawing, which I've just taken a look at. It's this absolutely brilliant and very, very sassy mosquito there. Thank you very much to Jude who sent that in. Really brilliant. Do send those in, anyone else who would like us to see them. That email address is in the chat. It was engage@welcome with 2 L's connectingscience.org. So what do we have for our last 2 sessions of this afternoon? Well, the first one, as I said before the break, is DN Apple code with our scientist Max Brown. Just before we get to Max, we showed you a couple of the techniques that we use to collect those life forms, mainly insects, before Petra's session. I'm going to show you another video now, which is what happens once we've collected them to get that DNA code out so that we can read their genomes. And you'll see lots of different bits of kit from our lab in this next video. Tree logo there. Thank you very much for that video, which came from the brilliant Luke in our team. So yes, you saw some interesting things there. Liquid nitrogen, power masher to break that sample up, and yeah, some other bits, centrifuge, vortex, magnetic beads, lots of exciting bits of equipment. You're going to hear a little bit more about those in this next session, DN Apple code, and of course get to do some brilliant activities. So I'm going to hand you over to Max Brown now for this next session. Thank you, Max.
Hello, everyone. Welcome to my session where I'll be talking about apples. And I hope you really enjoy the activities that we have planned for everyone today. So what we're going to do, we're going to look at our apples, sniff our apples and taste them. We are going to really try and notice how different and diverse apples can be. And we are going to do another activity looking at the code, the DNA code that was mentioned earlier in the afternoon. And we're going to talk a little bit more about the story of apples and where they come from. But first of all, we are going to do our apple investigation. So if you do have your apples with you, now would be a really good time to get them out. This is my one. It's a really lovely shiny apple. And so the first thing we're going to do is we are going to look at our apples, but we're not just going to look at them. We're going to really observe closely all of the different characteristics of the apple. So first of all, hold your apple like this, because this is how it would be growing on the tree. So this is the top, and this is the bottom where the stalk is. This apple is, the variety is called Spartan. It's a round shape. So yours might not be round, but this is a very, very round shape. It's got a little bit of ribbing, and it's actually got 5 little bumps at the apex, which is hard to see, but check that on yours. The skin is really smooth and shiny. You can even see it's shiny. And at the bottom of the apple, it's actually streaked. The color of the apple is probably the most obvious thing on this apple. It is a deep, deep crimson red. And it's a beautiful color. The stalk of the apple is the next thing to notice. Mine is quite thin, but yours might be really, really thick and stubby. And the very final thing to notice on the apple is the eye of what's called the eye of the apple, which is at the top here. And you should be able to see 5 little brown triangular structures called sepals. And we'll talk about those more in a bit. But making observations like this is a really important part of the scientific process. And I would invite you to write down your observations if you can. So the rows in this table are the different characters that you can see on your apple, like the stalk and the color and the shape of the apple. And if you do have 2 apples, you can go ahead and write your observations in for apple 1. And then if you do have another apple, which I'll talk about in a second, you can do that for the next one. So what can we notice about our second apple? Maybe you can discuss with another person in your room more about this apple. I've got another one here. Obviously very different is its color, but we'll go through the points which are on the slide. So first of all, look at the stalk. This one is quite well deep within the cavity, the bottom of the apple. What else do we have? The color of this apple is green. And Jack, our host, mentioned that he had a russet apple, which is what this one is. And you can notice that it has this sort of brown, rough texture to it. And that's almost like a corky, it's got a corky feeling to it. It's a really lovely texture. This apple is a bit different to the other apple. It's much flatter at the top and the bottom. And that's quite an important characteristic when we're looking at different apples. The eye of the apple is also very different. You can see that the sepals are much longer and they spread out more. So that is also another very important thing to look at. But it's important to remember that apples actually start off as flowers, which is really amazing to think about. And so you can see in the picture here, in the picture here, don't worry about all of the writing, but you can see where it says sepals, and that's kind of beneath the petals of the apple flower. There are these green structures called sepals, and actually they're the same sepals that you can see at the top of the apple here. And so you can really begin to relate the structure of the apple to the structure of the flower, which is really an interesting thing to do. Where it says ovary, that's where the seeds are, and around the ovary is a part which expands after the apple has been pollinated, and it expands and becomes really fleshy and delicious in what we now see as the apple. And in the core of the apple, that's where the pips are. So I've got a picture here of what apple flowers actually look like, and they're really they're quite large, and they flower mainly in the spring and early summer, and they are mainly pollinated by bees, so they're really good to have around if you want to keep the bees happy. So what are we going to do next? We're going to smell our apples, right? Now this is getting really exciting now, because we've done our observations of what it looks like, but now we're going to go through some other characteristics which are really important when we're thinking about apples. So give it a smell. What does yours smell like? Is it sweet? Is it really scented? Does it have no smell at all? Some apples don't smell. Mine is really sweet smelling. You might catch me smelling it over and over again. And the last thing we're going to do is taste our apples, but don't taste them yet. I'm just going to talk you through a couple of things which you might want to think about when you're tasting your apple. Think about the skin. Is it thick or thin? In this apple, it'll be thick and dry, and this one, they're quite thin. Is it shiny or greasy? Is it gritty? And then I want you to think about the texture of the flesh of the apple. So once you've bitten through the skin, you'll get to the flesh, and this should be the tasty bit, right? Is it crunchy or is it cottony? Is it crisp? And then once you've got to those, once you've gone past those textures, can you talk talk to each other in the room about what it tastes like, because that's probably the most important part of an apple, is what it tastes like. And is it sweet or sour? Does it make your cheeks tingle when you bite into it? Okay, have a bite and discuss what you think it tastes like. I'll give you a couple of seconds. I won't eat mine because I might need these later. Okay, so we'll move on. And as I hinted at before, and you can probably see just by looking at your two different apples, that they're quite varied. And actually, that in the UK, there are over 7,000 different kinds of varieties of apple. And so many that you can't actually fit them all into a single book. This book here is probably the most comprehensive book you can buy in a shop, but it doesn't have nearly all of those in there. In fact, some of them, here's another book. This book contains apples which were thought to be extinct, and people have rediscovered them in orchards, and they've been drawn and recorded in this book. So there's a lot of diversity out there. What we have been doing by looking at the shape and the smell and the taste of the apples is how traditionally people have studied apples throughout history. And if you liked an apple and you liked the way it tasted, you'd try and preserve that apple as much as you could. Or you could breed apples together with characteristics that you liked in the parent trees. But nowadays, we have other tools which you're probably thinking about, the topics which have been discussed. We can use other tools to help us find connections between apples that you can't see using your eyes. And this is where DNA comes in. I know you've heard about this earlier in the afternoon, but we'll quickly go over how this relates to apples. So, when we sample apples for their DNA, we take a leaf and we crush it, we freeze it, like you saw in the video, in liquid nitrogen, and then we crush it up, and it makes this sort of green paste that you can see in that image there. And then through a series of steps, you eventually can dissolve the DNA of the apple out. And this is what apple DNA looks like. It's a series of letters, G, T, A, and C, in a long, long line. And when we extract DNA from our apple leaves, it gets broken into loads and loads of tiny bits. And we have to try and piece them back together if we want to try and understand the original sequence. So we have to try and find out where the sequences overlap. And so here you can see that there are 4 sequences, and they overlap in differing lengths. And you get a sequence at the bottom, which is a long version of the overlap sequences. So we're going to have an activity now where we're trying to crack the apple code. Just to walk you through an example really quickly, there are 3 sections here. They're not in the right order, but we're going to try and put them together quickly. You might try and think about what goes at the start of a sentence and what happens at the end of a sentence. And if you look at them for a while, you might be able to see the overlaps. And we see that this spells out the sentence: an apple a day keeps the doctor away, which is a phrase we should all keep in mind. Okay, so you've been sent out this puzzle. I'd like to invite you to give this a go for 5 minutes and see if you can work out where the overlaps are, and see if you can unravel a part of the story of the apple. So I'll give you a few minutes now to to try and piece that together. If you don't have printouts of the sequence, the sequence of letters shown here, you can look at this screen for a bit. It will be a lot harder, but see if you can try and work out where the overlaps are, and see if you can try and find out what the sentence, the few sentences it spells out say. It might be helpful here to write them down. And once you've done that, you can give us an email and send in your answers, so we can see who's got the story of the apple right. To make it a bit easier, you might want to think about what happens at the start of a sentence. There might be capital letters. What happens towards the end of a sentence? There might be full stops. So you can see in the example here, there are 3 full stops. So that might give you an idea of how many sentences there are. If anyone has any Apple-related questions, please throw them in the chat. Happy to answer anything that comes up. Okay, should probably move on now. Let's see if you got it right. And we'll try and see if this gives us a hint as to what happens in the story of the apples. Okay, so the order was 5, 1, 2, 3, 6, 4, 8, 7. And it says, apples originated in Central Asia. They traveled west with people along the Silk Road over the past 3,000 years. Well, that's quite an amazing claim for something which we think of and eat every day in the UK and all around the world, actually. This is a Canadian variety of apple. So let's think about that a bit more. Apples have not been big and tasty throughout their history. This is an example of an apple which is at its full size, but it's tiny. And actually, these are called crab apples, and they occur in many parts of the world, and there's a wild species in the UK. Crab apples and things related to crab apples, which occur in the east towards China and the Tian Shan Mountains, are the original apples. Here, one species in particular occurs called the Central Asian apple, and it already has quite a big fruit and looks like a modern apple, but it isn't. And how did it get that big? Well, scientists think that the fruits are so large because large animals like bears began to choose the largest and sweetest fruits to eat. And in turn, wild apples were able to disperse and the seeds pass unharmed through the animals after they've finished with them. So actually, maybe we have bear poo to thank for our apples. The apples traveled west along the Silk Road, which is an ancient trade route. And it's thought that horses helped this eating and carrying people west from China. And so once it arrives in the West and Europe, there's this huge diversity of apples, because this apple, which originated in China, has bred with many local species of apple, and it creates this huge diversity. As I said, 7,000 in the UK alone. And so what are we doing at the Wellcome Sanger Institute? we are sequencing the genome of many, many different apple species, apple varieties, sorry. So we're trying to find out what parts of the DNA or the genome controls things in the apples like color or sweetness. So actually, there are parts of the DNA which cause the difference between this red coloration and this green coloration. And we might be able to find that out by sequencing our apples. And actually, we can unravel the story of apples even further, try and understand the details of how, when, and why apples are where they are now. So with that, I'd like to thank you for helping me and the team crack the apple code, and I hope you've had a nice time.
Brilliant. Thank you very much, Max, for taking us through those apples. And yes, I hope that you enjoyed piecing together those different bits of code. If you didn't get to finish it during our session today, then do see if you can finish off that printout afterwards. If you don't have the printout, then do just send an email to engage@welcomeconnectingscience.org and we'll get that sent out to you. That exercise, of course, piecing together that apple story in just the same way that we piece together those DNA sequences. We are going to move into our last session of this afternoon. I hope that you've enjoyed everything so far. This last one that we've got coming up is, as I said before, my favorite name, Spotty Dotty Squirmer Wormer with Erna. This is going to see how we use those codes when we've got them. So I'm going to hand over to Erna now, who's going to tell you all about a particular type of squirmy worms. Over to you, Erna.
Hello, everyone. I hope you can hear me. So my name is Erna, as Jack said. Thank you, Jack. I'm a researcher at the Tree of Life. So today I'm going to talk to you about the animals I look at, which are squirmy worms, tiny little ones called nematodes. And we're also going to use an art technique that we learned in homeschool that many of you might have done too, called Pointillism, to show how nematodes vary from place to place in different types of habitat. And we're also going to find out about how studying nematodes can also help us protect the planet, even though they're really tiny. So, So the keywords are going to be nematodes, which are these tiny worms. This is one here. This is C. elegans. It was the first animal to have its genome sequenced. So we'll talk a little bit about nematodes. We're talking about pointillism, which is the art technique. And we're going to talk about biodiversity, which is the variety of life that's on Earth. Abundance, which is the number of an animal that lives somewhere. And the community, which are the different types of animal that live together in a place. So about me, I'm going to talk about me. So this bucket kind of represents both my home life and my work life. I'm a mum myself of 3 kids, and they love building sandcastles, digging in the beach, that kind of thing. But also in my work, I use a bucket to collect sand, and then I look inside the sand to see the nematodes inside it and identify those. So my background is in ecology, which is where we look at the animals that live in a place and the place themselves and the relationship between them. And I used to do that by looking down a microscope, but that takes a long time. It's very difficult, and you can only do certain types of nematodes that you learn how to identify. So I then have veered away from that into using genomics, and I'll talk about that in a moment. So, sorry, this is very slow. There we go. So your first job now is to draw a worm. And I would like you to draw a worm around about the same size that you think a worm is, and the different body parts that you see, you imagine and remember seeing in a worm before. So I'll draw one quickly myself, actually. I should do that. So I'm guessing that you've drawn something. I'm not like Petra. I'm nowhere near as good as Petra, so you'll have to forgive me. But I'm guessing you've drawn something like that. Have you drawn something like that? And that is an earthworm. So if you can see there, it has little lines along its body, and those are different segments, and it uses those in moving, that kind of thing. But the worms aren't just like this. They're all different types of worms, and these are other types. They're different shapes and sizes. There's some that are predators that eat other animals. There's some that filter feed from the water around them. There are some that are parasites. But the type of worm I'm going to talk to you today is about nematodes. So this is a nematode that I picked up in Essex a few weeks ago. The name nematode sounds funny. It means thread-like. It's from the Greek words. And they're called thread-like because of their appearance. And if you look at this scale bar on the right-hand side at the bottom, this is 0.5 millimeter. So you know, you have a ruler, hopefully some of you have a ruler, and you know that you have these different measurements called a centimeter, and the two of these scale bars together would be the teeniest, tiniest measurements that you have on your ruler. It's a millimeter. So this nematode is about the same size as the smallest measurement, less than, it's 1/10 of a centimeter on your ruler. So they're quite tiny. And I've called that because of their, we're having technical difficulties. So what you're, here we go. Okay. So yes, these are wriggling nematodes, and that scale bar, if you can see here, this is a Petri dish and the scale is about 1 centimeter. So can you see that square within that? That's about the same size as this screen is. So I hope you're supposed to be able to see wriggling worms, just to have the bit of the wriggling in the squirmworm title. But each one of those nematodes is like a tiny speck of dust. If you line 10 of them alongside one another, they'd be about 1 centimeter between the two of these numbers. So, but you'd only see them as this tiny wriggling speck of dust. So I don't think that video is going to work. We'll just move on to the next slide, hopefully. But nematodes are basically everywhere. There are 57 billion nematodes for every single human on the planet. There are about 8 out of 10 multicellular or complex life forms are nematodes. So they're not just the most common type of animal. They're not the most common type of worm, they're the most common type of animal. And their success, their ability, how they've spread so far and wide, is mostly down to the different diversity, the different types of way they can get food. So these are a few of their mouth parts here, and you can see here this top one here has this little structure. It's called it's a stylet, but it's used to like a straw to suck from the roots of plants. So that's a parasitic nematode. Then you've got the rest of them are all free living. The other ones are ones that I've worked on. So this one, and this one are all different types of predators. So they eat other nematodes. They eat small other animals, multicellular animals like oligochaetes and different types of life. And then these two here feed on bacteria. This feeds on, it's an epistrophe feeder, feeds on different algal cells. And so they're different types of feeding. So that's helped them exploit lots of different habitats and spread far and wide. So nematodes are basically everywhere, and they're in every type of habitat, from the driest desert to the coldest habitats in the Arctic. And sorry, there we go. And yeah, so they're everywhere. They're hugely diverse. They reckon there's somewhere around 1 million species of nematodes. Some people estimate it up to maybe 100 million, which is a lot of different types of species. Sorry. So this is a quote I love by a man called Nathan Cobb. And he said about over 100 years ago, if all the matter in the universe, except for the nematodes, were swept away, our world would still be dimly recognizable. So if you took everything away in the universe, except for the nematodes, you'd still be able to see what was there. The mountains, the rivers, the river, the sea, the soil, and even to the extent of seeing the animals that were there and the plants that were there, because you can look at the parasites and tell what species of animal and plant was there, which I quite love. So the ones I work on are all free-living nematodes, but not all of them are. So as I said before, you have some parasitic nematodes. So these ones here are, this here is a carrot that's been plagued by this root knot nematode, which which parasitizes the carrot. But some parasitic nematodes have been quite useful. So they can be used to combat slugs in your garden, for example. So this is a slug killer, which you just put down these nematodes and they eat the slugs in your garden. And there's some that eat on other ants and things like that, and then some that feed on leaves inside plants. But the ones I'm going to talk to you today are about nematodes, and we're going to talk about how they can be used to protect the environment. So it's okay to collect nematodes and to use them in environmental quality monitoring because they are everywhere. they're so ubiquitous, it means they're absolutely everywhere. So you're not going to damage the population by taking a scoop of mud, and you can identify the species there and look at the diversity. That means how many different types there are. And then if there's lots of diversity, it tends to mean that you've got good environmental quality. And if you have lower diversity and you have certain species, it tells you something about the habitat and how good the quality is. So next thing I want to do is draw a little nematode. Maybe choose a different color and have it right beside your drawing of your worm, and just try and draw the smallest little speck next to your worm. And that shows you the different size scale of a nematode versus an earthworm. And they're really, really, really tiny. So far we've talked about how nematodes are a very small type of worm. They're very common, live in so many different types of places, and there are different types of nematodes, like different types of place to live. So you'll find different types of worms in your garden than you'll find on the beach. So now we're going to move on to pointillism. Pointillism is an art technique that we did in homeschool, which was a fun time. I'm sure that many of you who have children who are in year 3 during any of lockdown will have covered this a little bit. This picture might look a little bit familiar. This is by George Surratt, and he's the person who really invented pointillism. And it's where an artist used small dots of color to form an image. So it was kind of called a scientific style of painting at the time. And if you looked really closely at this painting, you would see that the green grass that we see at this distance is actually made of lots of different colors. They have orange dots, blue dots, as well as green. But from far away, the dots merge together to form this image. And we're going to use that technique. I thought it would be really appropriate to illustrate Cobb's quote, because if we think of every dot as being a different nematode and every different color is a different species. So yes, I like this bit. He said, trees would still stand in ghostly rows representing our streets and highways. The location of various plants and animals would still be decipherable. And in many cases, even their species could be determined by an examination of their remaining nematode parasites. So now we're going to do a pointillism picture using nematodes. So first of all, I'm going to very quickly ask, how many different types of places can you spot in this? And just have a chat with whoever is with you. So when I asked my son this, the first thing he spotted was the forest. So that will have its own community, which I've colored in green here. Different types of nematodes community would be there, but they'd be similar across different types of forest. And then the next one you might have spotted would be the marsh, which is that kind of grassy looking place. And then the sea one, the marine habitat, which is all the blue dots there. You've also got the fresh water, which is in the river. And then this really tricky one, but in between where the river and the sea meet, it's a special kind of habitat called an estuary as well. So if you got that, bonus points. So there's 5 different habitat types there. And each different habitat has its different community. So I'm going to go back to my bucket, and we're going to look at the number of species. So let's go collect some sand. And in the sand, we're going to start looking for nematodes. So if you look first, you'll find the most common type of nematode. We're going to call him number 1. Look a bit harder, we'll find number 2. Look harder still, number 3, number 4. And look really, really hard, we find number 5. So in that habitat, we've got 5 different nematode species in that bucket. Then we go to a different beach, collect more sand, and we look at how many species we have there. And if you look really, 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 really hard, you might find that you also have 5 species, but it's not as even between the number of species and the number of individuals. So which is more diverse? Do you have an idea? I would say, I think you might agree, that this one is slightly more diverse because it's a more evenly distributed among the species. So it's a different measure of diversity, but you have the number of species and how evenly they're spread amongst the species. So the first one I'm going to do quickly is this habitat here. So I'll start, and you're welcome to join in if you like. So if you were to look at this beautiful beach and wanted it, how many nematodes might you see? Well, we're going to start with the palm tree, that lovely coconut palm in the middle. And coconut palms have lots of different parasitic nematodes, and they are quite bad for the poor little coconut trees. And there's one in particular called what's it called again? A red ring nematode, and he goes into the trunk of the palm tree, and they can actually kill a palm tree in like a couple of weeks if they're infected. And there's this kind of beetle, a weevil, that spreads it. So the beetle itself is a parasite, and he also carries with it these nasty nematodes that can then kill kill it quite quickly. So I'm going to use lots of different colors. I'm using red for the red ring, and I'm using green for the parasites that go in the leaves. And so there are actually 23 different, or at least 23. That paper I read was like quite old. But lots of different nematode species that just parasitize just the coconut palm tree. And then we've got different types of parasites, that live, and different types of nematodes that live in the roots of the tree as well. So we're going to go, those are going to be a different color. So this is what I've drawn so far. I should have used that fancy camera that Bet was using. So this is what we're doing lots of different dots where each color is a different nematode. Each dot is a different nematode, and each color is a different species. And I'm going to do brown here for the soil nematodes as well. So maybe some green is going to be the next most common type of soil nematode there. And then we're going to do the next habitat I'm going to do is the beach. So I'm going to do the most common species there. I'm going to do is yellow. And we'll do lots of yellow dots here for the sand. And the beach itself has high diversity. It's got lots of different types of species that live there. And also kind of fairly high abundance. That means lots of individuals there as well. So we're going to do that's yellow. And so we're going to do a few different types of species. So let's do a few different colors here. We're going to try and do fast dots. And I should also say, if you're going to be dotting with your markers, to perhaps go gently on them, because we had some accidents in my house earlier with them dotting and markers, and I don't want all your markers to be ruined. So we're going to do then we're going to move on to this subtidal nematodes. This is the nematodes underneath the sea, and you have fewer dots there because the abundance is a bit lower there. So you're going to have fewer, but they have the same species. So we'll do some of the same colors as well. And there's some species that are only under the sea as well. So this is just to give you an idea. So this is one I'm just going to show now. I'll leave it at that because I don't want to take up too much of your time with me drawing, but that's the idea. So the different dots are different nematode species. And we've got the red one there is that parasite of the coconut palm. And yeah, so that's a quick indication of what we're going to do, what hopefully you're going to do with me next. So now we're going to do it together. Now, you can do this on a different picture if you like. If you have a fabulous place that you love to visit or anything like that, please feel free to use that instead. But the one I'm going to do is going to just kind of link it in to environmental quality. So it's not as pretty a picture as the last one, but it's kind of important because this here shows the impact of disturbance. So this big long tube is actually sewage treatment, outflow. So it's where there's a lot of nutrients going into the sea, which you probably don't want there, and this has a bad impact on the environment quality. So you can see here there's a lot of green, and that's all because there's too many nutrients going in here. So you'll have a big impact on the diversity, you'll have lower diversity, and you'll have some of these species that show us that it's bad quality and it's polluted. So if we were to do that really quickly, I'll just start now, a different page. So we're going to do green for the ones that are most common along the pipe, along the strand up there. And then we've got people there as well. So we're going to do something with those people as well, because remember what I told you about parasites and the dog as well. So we're going to do the green for the most common kind of algae you see the green. So they're going to be different types of green. We're going to do different types of green for that. And we're also going to do the sand. Let's do the sand next. We're going to draw yellow is going to be the most common type that we see on the beach. So that's going to be quite lots of dots. If you can do lots of dots quickly, we're going to do lots of dots for this because they're quite abundant. But also, if you can do lots of different colors, I'm going to do orange for our next sand free living nematode. And some of these will go into this under the water as well. So we'll do some of those dots up there as well. And then we're going to I'm going to do some parasites. I'm going to do some parasites here. So I'm going to do those in black. And I'm going to draw the dog, because dogs have different types of parasites that sometimes can go to humans. So I'm going to draw one of those humans, hasn't washed his hands very well, or their hands very well. So they've got some parasites as well. And then what else are we going to draw? Oh, now we're going to draw the indicator species. So because there is too many nutrients, this system has this indicator species, which tells us that it's not very good quality. So we're going to do them in red, and we're going to show that where the nutrients go out, we've got lots more of those. So I'm going to draw the ones that the species that you only find in the water. I'm going to do those in blue. They're going to be only where the water is. They're unique to the water and you don't get them on the beach. Okay, so that's really very quick, and I'm sure you can carry on drawing as I'm talking you through the next bit, and I'll show you my final one that I did earlier at the end, and you can show me yours if you want to send them in. So this is kind of roughly mine. You can't see it very clearly, but there's the parasite, there's the dog and the person. These are the green algal feeders there, and then you've got the beach, the yellowy ones from the beach, and then the blue ones from the sea, and then these red indicator species. So yeah, I'll show you the one I did earlier later at the end. So you can carry on drawing if you want. You don't feel you need to stop. So how can we use this to protect the environment? First of all, we can use them to protect environmental quality, because if you look at the species that you have, you'll have fewer species where you have pollution. So, and we'll also get these indicators, so they can be good or bad, and they'll tell you something about the habitat quality. So these are some examples. This one here, they've got these beautiful structures on them, which we don't really have time to talk about. But these two here on the left, those indicate good environmental quality. They would say to you, if you found them, that you don't have any nasty heavy metals in your habitat, and that it's okay. The food chain around it is very safe. If you're going to eat food from around there, it's okay. And these ones here on the right, they tell you it's not such good environmental quality. This one here, you can get that. You get a lot of those where you have some heavy metals in your system, and you probably don't want to eat food from around there. And this one here kind of means, on the right hand side, means you've got organic pollution. So yes, maybe too much sewage or runoff from farmland or something. So they can be very useful to tell us about the habitat quality. So you can look at that two different ways. You can look at it morphologically, where you look down a microscope and you look at the nematodes and you identify them by their mouth parts, by their cuticle, that's their skin, and the different kind of features like that. Or you can look at it with their DNA. Not managing to move on. So how can using DNA help? Sometimes animals can look identical, but they're actually different species, and that's called when they're cryptic. And you can only tell them apart using their DNA. They can't reproduce together, so they're different species. So DNA barcoding is a faster way to identify nematodes than to look at them down a microscope. So it took me 4 years or thereabouts looking down a microscope to do the same work, to do my PhD, my studies, and then to look at it down with their DNA. It took about a month or two, so far quicker. And it was actually reflected quite well, the results that I did by looking down a microscope. So that's why I'm kind of moving away from that. It's a lot faster. So these, as Charlotte and Sophie said earlier, all different types of life look very similar, but they're different. If you look at the code, they can be quite different, but have similar code, just a different order. And so we use that when we're looking at using DNA barcoding. So first of all, let's start off with our bucket of mystery nematodes. And we don't know what they are, but we're going to take all of the nematodes out and extract their DNA, and we'll put it into a little tube like this one here, full of mystery nematode DNA. How do we figure out what was in that? So a lot of different people have spent time looking at nematodes and sequencing their DNA, so figuring out that book that represents that animal. Now, for nematodes, they've generally done it with a short little bit of their DNA to tell apart the different species. So we can take our little bits of DNA and we use it against these known bits. So we've got mystery bits, and we compare that to these known sequences where somebody has put it all and it's made it available for everyone to see, and we can match it against it. And once you can match your mystery sequence to a known sequence, you can figure out what species it is. And then once we've done that, then it's not such a hard thing then to see if you have these indicator species. So if you have, for example, species 3, and you identify him as being Allodiplogaster paraarmatus, you know that you have organic pollution. So that's how we can use DNA barcoding of nematode species to protect against, to look at our environmental quality. So today we've learned a bit about nematodes, pointillism, biodiversity, abundance, and the community. And I hope that you've enjoyed your session here. And I'd really like to thank you very much for staying on this long, and thank you to everybody. And I'll pass you back now to Jack. So thank you very much.
Thank you so much, Erna, and I hope that you've all been doing some brilliant nematode pointillism for us. There's some great words in there, isn't there? Thank you also very much to Erna, who went through that at lightning speed to ensure that we got through to finish at 4:30. Thank you hugely, Erna, for that. Yes, please do send in any of those nematode pointillism artworks when you have them done. That's to the same email address, which I think is just on the next slide. Erna is showing us her pointillism. Let me turn my camera up. Come back, Erna, come back. Let me turn my camera off.
Sorry, guys, I just forgot to show you my picture. The girls reminded me. So there we've got the red ones here around the end here, lots of them around there. And they're our indicator species telling us we've got nasty organic pollution there. So too many nutrients. So that's the idea. And I would love to see your pictures if you wouldn't mind sending those in. So anyone who managed to stay on, thank you very much. Sorry, Jack.
Oh, I'm really glad that you showed us that. It's a beautiful picture, Erna. I need to see that in more detail. But yes, we've got our email address on there, engage@welcome with 2 L's, connectingscience.org. Do send in your pointillism pictures. I'm sure they look as beautiful as Erna's does. We're gonna wrap up the session today. It's been brilliant to have you on the call with us for this first Curiosity Club. Huge thank you to our scientists for all those brilliant activities. And I'm going to hand you back to Becky now, just to finish. Thank you.
Thank you, Jack. And yes, thank you again to Charlotte and Sophie, to Max and to Erna and to Petra for their fantastic sessions. Really different ways of thinking about such a big subject. All these things that make us, make all the living things around us what they are, but just thinking how we're all connected by that huge string of DNA. This is the first in our curiosity clubs with the pandemic. It's been a while since we've been able to see everybody, and we really hope that we can welcome you back to the Wellcome Genome campus again before too long. But we want to know what you think about what you've done today. We hope you've enjoyed it, but we want to know if there's things we could do differently, how we can make them better for you. So at the end of this webinar, there'll be a link that will come up that will take you to our online survey. And we'd be really grateful if you have a chance to answer some questions in there, whether that's straight away or you want to do it a bit later on. We'll also email out that link to you as well. For taking the time to fill that in, you will be, if you would like to be, entered into a prize draw to win a £50 Love to Shop voucher. So that could be useful in this build up to the Christmas season. So we'd be very grateful to know what you think, and we really hope to see you again sometime again soon. So enjoy the rest of your weekends. Bye.